Hi, everyone. This is part two of our discussion about contraction and relaxation of skeletal muscle cells. The learning objectives are similar to the previous video, but this time we focus more on the cross-bridge cycling and the sliding filament model. We will also briefly discuss how relaxation occurs in skeletal muscle. So here's a slide that we've already seen, and that's a reminder that we've already discussed steps 1 and 2. So we know that acetylcholine depolarizes the muscle cells at the neuromuscular junction. And we also know what excitation-contraction coupling is, that translation of action potentials into calcium signals. This video focuses on step 3, which is the cross-bridge cycling. In a nutshell, the cross-bridge cycling means that the calcium that is now inside the muscle cell allows for myosin and actin to actually interact together. And that's going to lead to the shortening of the sarcomeres and muscle contractions. So we finished the last video talking about calcium increasing inside the sarcoplasm. So let's compare and contrast what happens with the thin filament at rest, which is on the left side here when calcium is low, versus in a stimulated muscle here on the right when calcium is high. So just to know what we're looking at here, so here we're looking at the thin filament. So each of those globular proteins is a molecule of actin, which together forms the thin filament. And then we'll also see the associated proteins. So we see tropomyosin here in blue and troponin in brown, identified with TN. And as we know, at rest, the tropomyosin here actually hides the myosin binding sites on the actin. So as we can see here, the myosin head and the actin cannot interact, right? There's a space here showing that there is no physical binding of the two molecules, basically because tropomyosin is in the way. So let's contrast this with what happens in a stimulated muscle when calcium is high. Basically, calcium changes that situation. The calcium here is represented with those yellow dots. And you can see that when the calcium in the cytosol increases, it will bind to troponin, which we see right here. Once again, it's a story of conformational change, or change in the shape of the protein. So when calcium binds to troponin, that causes troponin to change its shape, or change its conformation. And as a result, this actually pulls on the tropomyosin. Right? Remember that troponin and tropomyosin are connected with one another. So if troponin changes and moves upward, it brings tropomyosin upwards as well. And what this does is now makes the myosin binding sites available. So we don't see them on this cartoon here, but I will add them in black here. So let's say these are our myosin binding sites. We can now see that the myosin can now physically interact with the actin and form those cross bridges. So this slide hopefully illustrates the importance of calcium inducing the conformational change of troponin, and that causes tropomyosin to move and then myosin binding sites to be revealed and available for myosin. So we've seen this slide before. The take-home message here is that during muscle contraction, sarcomeres shorten. And we see that here in a relaxed muscle, the sarcomeres are longer than in a contracted muscle. What is different here is that we add the name of this process. So this is called the sliding filament theory. So when the sarcomeres shorten, the length of the thick filament doesn't change, right? It's exactly the same. And the length of the thin filaments doesn't change either. What happens is really that those two filaments slide past one another. And we see that by the amount of overlap between the actin and myosin here compared to a contracted sarcomere. And now what we're looking at is obviously just one sarcomere, but think that in the myofibril, in a skeletal muscle, there's hundreds and thousands of sarcomere that are lined up one next to another. So when we have many sarcomeres shortening at the same time, the overall effect is a shortening of the muscle, which is what a muscle contraction is. If you think about doing a bicep curl, for instance, your bicep is longer at rest. And when you do the bicep curl and you contract your bicep actually shortens. And that happens because of the action of these sarcomeres. Now, the last question we have to answer here is what actually takes place between actin and myosin for actin to slide towards the myosin? And the answer is called the cross-bridge cycling. What it is rapid and repeated cycles of myosin and actin associated together and dissociating from one another. So they'll basically bind together, unbind, bind, unbind, bind, unbind. So this happens really, really quickly and can happen for a sustained amount of time. And that's what leads to the sarcomere shortening and muscle contraction. And as we know, this can only happen when the concentration of calcium in the sarcoplasm is high, because that's when myosin and actin can bind to one another. So it's a 5-step process, which we're going to discuss here and in the next slide. So step 1, or step A, is called the rigor state. In this state, the head of the myosin is bound to actin. Now, remember that when we discuss myosin head, we said that the head of the myosin has binding sites for two things. It has binding sites for actin, and it has binding sites for ATP and ADP. So in this case, myosin is bound to actin, so that binding site is used, but this is the only state where no nucleotide is bound to the myosin head. So ATP is not present and ADP is not present. In living muscle, this rigor state occurs for a very, very, very brief period because we have sufficient ATP available in our cell to quickly bind to that binding site. And that's what happens in step B, which is called ATP binding. So we had an available binding site for ATP or ADP in the myosin head, which wasn't occupied. Because there's ATP in the muscle cell, ATP will bind to its binding site. What this does is that this reduces the affinity of myosin and actin. In other words, when ATP is bound to myosin, it's harder for myosin to bind to actin just because of the shape of the proteins. As a result, the myosin head releases from the actin, and we can see that here. There's no longer this binding. Another way of thinking about this step is to say that ATP binding is required, and the key word here is required, for myosin to dissociate from actin. So that's step 2. Step 3 is called ATP hydrolysis. So let's recall another important concept here. The head of the myosin has an enzyme activity. It has an ATPase, which is an enzyme that can break down ATP. And it is active in that third step. So at that point, ATP is broken down into ADP and a phosphate. And we know that this reaction releases energy. So now myosin has energy available to do something. So what is myosin going to do with this energy? Myosin uses that energy to rotate the head of the myosin to the cocked position, which we also call the ready position. Let's look at the angle of the myosin head here. So you can see the hinge here, and then the myosin head is almost upright. Let's compare this to what we saw in the previous slide. The hinge is still there, but the myosin head is positioned at an angle here. So I can quickly draw this here. So in step 2, or step B, that we just saw, the hinge is here and the myosin head has an angle. Step C here, we still have the hinge region, but the head of the myosin, it's much straighter. So that's what we call the cup position or the ready position. And the difference between the position at step and the position at step C is really the hydrolysis of ATP. So the energy that was harvested from breaking down ATP is responsible for the rotation of the head to the cock position. At this point, because ATP is no longer bound to myosin, it's a little bit easier for myosin to bind to actin. So the myosin head and actin still interact here, but it's a pretty weak bond. So here's our fourth step, which is called the power stroke. The first thing that happens here is that myosin will release the phosphate that's bound here. We know that there's a phosphate molecule that was made in the ATP hydrolysis. This phosphate group dissociates from the myosin. And this transforms the bond between actin and myosin from a weak one to a very strong one. So now the cross bridge here that we see is very, very strong. At that point, the head of the myosin swivels. So it rotates a little bit. And you can see that by comparing, once again, the position of the head here to the position of the head here. And what this does is that it brings the actin a tiny bit closer to the middle of the sarcomere. And that's how the thin filament slides past the thick filament, as we just explained in that sliding filament theory. And our fifth and last step is ADP release. So at this point, ADP will be released from the myosin. This increases the affinity between myosin and actin, and we're back to the rigor state, which was our first step, and a new cross-bridge cycle can start. So here's the overview of that cross-bridge cycle, right? We started at the top here with the rigor state. The key characteristics that you need to know about these steps is what is bound to the myosin head, the position of the myosin head. Is it in that cocked or ready position? Is it swiveling? We also need to understand the role of ATP and ADP in the myosin's head action, as well as the strength of the bonds between myosin and actin. So let's recap here. In the rigor state, there's no ATP and no ATP bound. That's a tight bond, so a strong bond here. But that happens only for a very, very brief moment in living muscles. Because as soon as ATP is available, and it's always available, ATP will bind to the myosin head. And as we know, ATP decreases the affinity between myosin and actin, and myosin detaches from actin. Immediately after, the myosin ATPase in the head will break down the ATP and convert it to ADP and phosphate. That released energy that is used to reposition the head of the myosin in its cocked position. Fourth step is power stroke. First, phosphate is released from the myosin, which increases the affinity from myosin to actin. So the bond here is very, very strong. And then the head of the myosin swivels, pulling the actin toward the middle of the sarcomere, making the sarcomere a tiny bit smaller. And finally, ADP is released from the myosin head. This increases, once again, the affinity between actin and myosin, and we're back to step A. Obviously, each of these power strokes is very, very small, so we need a significant amount of these cycles in order to get a meaningful muscle contraction. One of the implications of that is that muscle contractions require a lot of ATP. And in the next video, we'll actually talk about where that ATP comes from. So we've now answered the question, how does skeletal muscle contraction occur? And we have these three steps. First, the events at the neuromuscular junction, then the events of excitation, contraction, coupling, and then the events of the cross-bridge cycling. The next slide talks about how does skeletal muscle relaxation occurs. And the quick answer is that calcium must be removed from the sarcoplasm. So how is the skeletal muscle cell able to decrease the concentration of calcium inside the sarcoplasm? There's basically a two-step answer. So at first, the muscle must stop getting stimulated. So all of the activation events that we talked about must be stopped. So the motor neuron will stop releasing acetylcholine in the neuromuscular junction. This allows the muscle fibers to depolarize, and therefore the RYL channels close, and then the sarcoplasmic reticulum stops releasing calcium. So that's part of it. However, that is not sufficient. And that's where our second step comes from. The muscles need to also move the calcium that's in the sarcoplasm back to the sarcoplasmic reticulum where it belongs. Now, the challenge here is that we know that the sarcoplasmic reticulum has a very, very, very high concentration of calcium. The concentration of calcium in the sarcoplasm is still lower than what it is in the sarcoplasmic reticulum. Therefore, if we want to get calcium ions from the sarcoplasm back to the sarcoplasmic reticulum, so in this direction, we must use active transport because calcium is transported against its concentration gradient. And that's exactly what happens here. So I want to draw your attention to this molecule here. It's a calcium ATPase. So we're very familiar with our sodium and potassium ATPase. This is a similar type of protein, but instead of transporting sodium and potassium, it transports calcium. So the muscles use these calcium ATPases in order to actively pump the calcium back into the sarcoplasmic reticulum. And this is really the key event that will allow the calcium inside the cell to decrease, and then the calcium unbinds from troponin. And then all of the events that we talked about just reverse. Calcium unbinds from troponin, tropomyosin slides back into its resting position and blocks the myosin binding site, and as a result, your muscle relaxes. Okay, the last two videos have been filled with information, so let's try to summarize this. So muscle cell is also called a muscle fiber. What are the contractile units of skeletal muscles? They're called the sarcomeres. Sarcomeres contain myofilaments, actin and myosin. Actin is the thin filament which is bound by two other proteins, troponin and tropomyosin. And the thick filament formed by myosin has binding sites for two things, actin and then nucleotides, so either ATP or ADP. So during the contraction process, the skeletal muscles get depolarized by motor neurons at the neuromuscular junction. By which neurotransmitter? We all know it's acetylcholine that binds to and opens the nicotinic ion channels. This brings sodium in. We also know that it brings potassium out, but what is really important for this process is the sodium influx, which then creates the end plate potential and the action potentials later on. And now we have action potentials conducted along the sarcolemma, and we know the sarcolemma isn't continuous. It has these dips that we call the transverse tubules or the T-tubules, and they link the sarcolemma with the sarcoplasmic reticulum via two proteins, the dihydropyridine receptor, or DHPR, and the ryanodine receptor, or RYR. Skeletal muscles contract using cross-bridge cycling, which requires calcium from the sarcoplasmic reticulum to bind to troponin. It also requires ATP. And the functions of ATP is to release the myosin actin bound and to reposition the myosin head. And finally, during skeletal muscle contraction, ATP is used to pump calcium back into the sarcoplasmic reticulum. Right? That's a type of active transport. There we go. So hopefully this lecture and this summary slide helps you put everything in perspective and help you deepen your knowledge of what goes on inside the skeletal muscle cell during contraction and relaxation.